not going to. So they diffuse to adjacent tissues, and as I've indicated, the primary effect, the one that you need to be familiar with, the one that you see and hear advertised just about every night if you watch network television, public video. What is the most common advertisement you see related to healthcare on TV at night?
Viagra?
Yeah, exactly. It wasn't really, and we all know what an urgent health issue that is around the world and how many lives are saved. But you get the idea that that's dependent upon these arginase, nitric oxide pathways, and vasodilation of critical. And these are really non-specific vasodilators. That's why they say on the label, you hear on the advertisements, if you have one for more than 4 hours, you're in trouble, because that's again, non-specific vasodilation. So we'll talk more about these. Key things to remember, I just want you to know that the gaseous neurotransmitters do not have receptors and are released and act very, very rapidly. There are nitrogenous bases. We sometimes call them nucleotides. For example, the genetic code consists of these nitrogenous bases: adenine, cytosine, thymine, and so on. These are the nitrogenous bases that make up the genetic code. But some of these are also act as neurotransmitters centrally. One of these is adenosine. It's a variant of the adenine, and it seems to also play a role in central diminishment or reduction of pain. And of course, a lot of these studies, it's hard to do pain studies. I mean, would you want to be in a pain lab? I used to walk by the pain lab at USF when I was in graduate school, and they always kept the doors shut, and just wondered what went on in there. But they did a lot of the awful things, I'm sure. I mean, of course, they worked on animals, and it's my knowledge they didn't work on graduate students so much. I didn't want to find out. But how do you know if the mouse is in pain? For your perusal, I submit the mouse does steal. Coding the facial expressions of pain in the laboratory mouse. Because they make little noises, they make happy little noises that we've now learned to pick up with, well, they make them in the ultrasound region. And now, in most behavioral phenotyping, the cages are equipped with these ultrasound microphones, and we monitor their little chirping noises, which you can't hear. But that helps too. But something to think about, because remember we talked about the issue of how do you know the state of mind of a mouse. You can't sit down and have a one-to-one conversation with them in most cases. All right, so that's all I want to say about neurotransmitters. If you have any questions about that, let me know. We also need to clean up a few other general concepts regarding the organization of the nervous system. And what we're looking at in this illustration are two patterns of signal propagation, neural signaling. On the left, we have something called divergence. On the right, we have something called convergence. And these terms will come up again and again as we continue our discussion of the nervous system. And in general, you can see what's happening. On the left here, in the process in the pathways of neural divergence, you have one cell giving a command or issuing action potentials to a larger number of cells. So it's one to many. In contrast, or conversely in convergence, you have a lot of cells funneling their signal to one cell. And this is going to be important, for example, in our discussion of the sensorium. We say, for example, that the autonomic nervous system, especially the sympathetic nervous system, is structurally divergent. And by that, we mean that you begin with a few cells issuing action potentials, and then those signals are then propagated to a larger number of cells. That's all that means. The sensorium, in some respects, is hampered by the fact that structural convergence is necessary. You have lots of receptors, for example, lots of rods and cones in the back of your eye, but boy, all that information and all those many rods and cones is then funneled into just a few axons that form the optic nerve. Other examples of this we'll talk about. Yes?
I'm having a little trouble understanding the divergence. So each
neuron only has one axon in one place for a synapse to occur. So is it communicating with all those other cells by the function of integration reaching out for that single axon?
When you say single axon, so for example, let's say this is a motor or efferent cell at the very top there. And you can see there's its one axon, but you can see it's branching axon. Right? It's a lot of motor neurons are like this. We'll look at an example of this shortly. So this one neuron, for example, is sending signals to 3 other neurons, same signal, same information. And let's imagine that these were muscle cells. All 3 cells would contract simultaneously, originating from one common signal. If they're neurons, they're simply going to receive the same signal, and that's about all we can say. If it's sufficient deep membrane depolarization to generate an action potential, then all of these cells would generate the action potential, which would then be communicated to these cells, and so on.
So the axons and brains often have different points of
communication? Yeah, that's very common in a motor or an efferent cell. We'll talk about the organization or the structure of these neurons in just a moment in more detail. I'll give you some clues about how to classify these. But more importantly, we're really talking about arrangements of multiple neurons and how signals are conveyed from one point of the anatomy to another. And remember an illustration of glutamate and GABA. When we first began talking about those neurotransmitters, we discussed something called summation. Can anybody define the summation? What do we mean by neural summation?
Yep. The stimulus of the cell is the summation of all the excitatory and inhibitory signals that the body receives. The action potential only occur if its summation is depolarizing at the point of the axon.
Oh, good. And so if you had many more GABAergic synapses than glutamatergic synapses, you would actually have net hyperpolarization of the membrane and less likelihood of a depolarization. So by summation, we're just adding up all. Remember we said that for any given neuron in your brain, there are probably about 10,000 inputs. 10,000 inputs, and those are then added together, both excitatory and inhibitory, and collectively then that postsynaptic neuron will either fire or not based upon the collective integration of those inputs. All right, so we sometimes make a somewhat theoretical, I would agree this is more of a scholastic distinction, between spatial summation of neurons and neuronal circuits and frequency summation. Bear in mind that both are really occurring in the brain at the same time. And in this illustration, you can see that we have a postsynaptic neuron here. Here would be our postsynaptic neuron, and you have 3 presynaptic terminals up there. All 3 are excitatory. I know that because I can see the little positive sign there. And so when all 3 are added together, that would be sufficient then to elevate membrane potential above threshold at the trigger zone. Now, if all 3 inputs, and here's the key thing about spatial summation, and again, why you must appreciate this is more of a theoretical art of this than a practical one. If all 3 signals arrive at different points along the cell membrane or on the dendrites simultaneously, and that's the key word, then that is termed spatial summation. So the key criteria in defining spatial summation is the inputs are at different physical locations on the postsynaptic cell membrane, and they arrive concurrently, they arrive at the same time. So the focus is upon the topology, the location. Now, let's look at this illustration on the left. The negative 70, you can see there. What does that suggest to you? Where have you seen that before? Resting membrane potential, typical RFP for a neuron. And then we have A1 here. How would you describe A1 to a 5th grader? Or maybe a 4th grader? Just explain it to me. What's happening here? Does a depolar, or maybe perhaps a graded potential, the depolarization, does it achieve threshold?
No.
So independent of whether we're at the axon hillock or not, it doesn't even achieve threshold. So we have some type of membrane depolarization here. We'll call it a graded potential. We have another one here. But the two together, they really don't have, they're occurring at different points in time. This is our time axis down here. This one occurs, then this one occurs. They have no effect on each other because they're so separated by the time interval between them. By contrast, what's happened over here? A1 has occurred, and before A1, before the depolarization is finished, before the RMP is restored, what happens? A2 occurs, and A2 piggybacks on A1. So this is called frequency summation. Frequency summation focuses upon the time axis. So in this case, it's not that you have simultaneous piggybacking and thereby the depolarization's added at the same time. Rather, it's a little bit off. It's not quite simultaneous, not quite concurrent. And so as a consequence, before the first one has a chance to completely decay, the second one occurs. And in this way, the threshold is reached, and again, if this occurs at the axon hillock, that action potential is achieved. So the key difference between spatial and frequency summation is that in spatial summation, they're all simultaneous. In frequency summation, not quite simultaneous, rather they're sort of time shifted a little bit.
So temporal and frequency summation.
That's correct, thank you. Yeah, temporal. So temporal just refers to time. Yeah.
So if you've got, say you've got the cell body, so you've got the soma, and you've got different places along that where different stimuli are coming in, and those are all graded potentials. So each one of those is going to take a different amount of time to get through. You know what I mean? So a smaller stimulus that's right next to the axon can add to something that's really large. You know what I mean?
Exactly right. So you would have to have very sophisticated instruments at the axon hillock and perhaps along the membrane to sort all this out. And as we've indicated, really what's going on with each of your postsynaptic neurons is both spatial and frequency summation are occurring. We just want to be aware that you understand that summation can occur with simultaneous signaling and with this sort of more time-dependent signal. Yeah, the proper term here is temporal summation.
And,
Yeah.
sorry, and the location matters too, right?
As George is pointing out, in reality it does, but in terms of temporal summation as an abstraction, we're still more focused upon time than upon location. Spatial summation is more important, or depends more properly upon the location. If you remember the illustration here, how would you describe what's happening on A there, in column A? Yeah, you have two independent depolarizations and graded potentials that have no effect on each other. I guess the E stands for excitation. Everybody see how this is temporal summation? You begin the first one here, the second one piggybacks on top of it. Spatial summation would go on C here. The implication is that this one and this one over on this side have now joined together. One, they've occurred concurrently, and in so doing, have exceeded threshold. And then what's happened here? Yeah.
An excitatory and an inhibitory are happening
Yeah,
at
and
the
the,
same time.
but be careful, we're quite at the same time, but what happens with the net effect is there's no propensity, there's no inclination of this member of this cell to generate an action potential because the net effect is to keep us at RMP. Yeah.
How does your analogy to the jumping relate to temporal summation?
Actually, when I was talking about this, it occurred, I was thinking about when I was about your age, taking water safety instruction in Maine. And if anybody ever taken water safety instruction, you have at one point they make you dive into the water to death, and you have above you a drowning person, and your job. I've never quite understood why you're asked to do this, but your job as the person saving or rescuing the individual is to dive down, grab them by the ankles, and throw them up as far as you can. Don't know what that achieves actually, because they're not going to, unless you can throw them off onto the shore or something. But what I found is that if I could nudge them like a porpoise closer to the shore, because when you shove the person up, you're going to go down and potentially drown yourself. I learned that lesson quickly. But if I could I would all come up, yeah, it's been forever, I'd be laughing. And I found that if I could move over toward the shore, when I pushed them up, I hit the and so if you extend that, suppose I was actually standing, I'm in the water, and I extend my legs down to thrust them up. All right, I'm going to get the initial force from being on a solid surface. You mentioned the jumping analogy. If you're jumping in the air, if you know anything about inertia, if then I jump on you and jump, I'm going to get a little extra height. You're going to go down by Newton's third law, but in so doing. So I'm not sure we needed to go there, but that would be the attempt I would make to use that analogy. Yeah, maybe.
So the temporal solution is not an example of a factory equation. What is it?
No, that would be a different notion. Here we're just talking about what's happening at the axon fillet. So temporal summation would just be almost simultaneous excitation of the membrane, depolarization, resulting in threshold being exceeded. Yeah.
So C, the excitatory 1 and 2 occurs at the same time?
Well, it would here. That's not shown here. This is you're supposed to, the implication here is let's move these over here and then do this.
So yeah, so those occur at the same time. But on the other one, on D, they don't occur.
Well, no, they would not only do they not occur at the same time, they effectively then they're trying to suggest that you cancel the effects. One cancels the effects of the other. You're left at RMP again. And this is just to remind you, I just wanted, we saw this before, but don't let all of these terms escape your attention. Make sure you know what we mean by an excitatory postsynaptic potential and inhibitory postsynaptic potential, and you can see the effects on, you can see how this all then relates to summation, and we talked about the various categories of summation. We mentioned, I think I've already asked you this question, this is all we're talking about down here. What are the two general ways we've talked about by which membrane potential can be made or rendered more negative? What? Yeah, chloride, that would be the GABA. What's the other way? Potassium channels all do. And so the other point that's being made here, not terribly elegantly, is again, I'm suggesting once again that you have to be careful with the terminology. I know some of you coming into the office talk about increasing or decreasing membrane potential. What's the problem with that, with the use of that terminology? If I go from negative 75 to negative 55, am I increasing or decreasing membrane potential? All right, we have a vote for decreasing. Anybody want to go for increasing? Increasing. Everybody going to increasing? All right. So why would you say it's increasing? Just find it out. Why would you argue it's increasing? Why would you argue that it's decreasing?
More positive, getting closer.
So you're moving more positive, and so that would be a depolarization. We're going from a more negative number to, well, actually we're increasing the magnitude in that sense. But why would you also say that you're increasing or decreasing membrane potential? That's right. You're moving closer to zero, and so we could talk about absolute value. And it's really, no one asked this question. I'm surprised in this group that no one annoyed me with this question. What about in an action potential when you go from 0 to positive 30 in depolarization? Is that really depolarization or?
Yeah.
Polarization.
Depolarization. Depolarization.
Yeah. All right. if it's, what is the definition of depolarization? We're removing the polarity across the membrane. And going from 0 to positive 30, are we removing the polarity across the membrane? If I go from here to here, this is 0. Yeah, you're increasing. So wouldn't that be polarization? Trying to get you to lose sleep tonight. That's not something I would ask you. I'm not surprised that no one, I usually get that question. We call that depolarization, and that's what I want you to think of it is. But in reality, you're because of this peculiarity of the numbers you're using in the 0, in effect, you're polarizing the membrane as you go towards the Nernst potential for sodium. But again, that's the only point of this slide, just to be aware of the terminology. If you look at this illustration right here, so you would agree that we have a presynaptic cell here. We'll call this pre, and we'll call these our posts over here. Okay. And you can see we have neurotransmitter being released there. So this little axon terminal here is actually juxtaposed or adjacent to a synaptic terminal, another presynaptic terminal. Does everybody see that? It's not, this thing is not over here on the postsynaptic cell, rather it's modulating or altering the behavior of the axon terminal. And that is called presynaptic, because we've just been talking about postsynaptic excitation, EPSP, IPSP, everybody knows? All right. This is called presynaptic inhibition or excitation. And we could quibble with the logic of that, but that's what the term is. Presynaptic. In this case, it's inhibition. It's diminishing the amount of neurotransmitter that's being released. And most of these presynaptic circuits are inhibitory. They generally decrease the amount of neurotransmitter released. So architecturally, organizationally, what defines presynaptic inhibition? Well, that's true, function, but in terms of the organization, the structure, how would you identify if I were to ask you about presynaptic inhibition? How would you identify it? By just looking at the arrangement of the neurons? All right, you have the terminal up there, the axon terminal, not juxtaposed to the postsynaptic cell, but targeting or modulating another presynaptic cell. This is one cell just basically interfering with the activity of another.
So those terminals have receptors for neurotransmitters to be released. That's what happens.
Yeah, it gets even more complicated than that, but yes, that's certainly true. Yeah, I could tell you stories about the organization of synapses that would curl your hair, but we don't need to worry about that. And so this is a little exercise. If you've gone to Burger King or Hardee's or whatever, these are all my favorite places, and they will give you a placemat on your tray, and you'll have to help find Waldo or something, help find your way out of it. So what I'm asking you to do here is find or identify all the mechanisms shown in this illustration by which the neurotransmitter is being removed from the synaptic cleft. So what do you notice here? You should see 3 or 4 anyway. All right, there's going to be reuptake. That's going to remove it. That would be this one. What else is happening?
There's an enzymatic.
Yeah.
Yeah.
When you think of maybe the enzyme, it's not quite the right location, but which enzyme did we say was away from that breaks down the amines away from
Acetylcholine.
CNP, the catechol-transferase. That might be it. You might have a receptor saturation that would tend to lower depending upon the ratio of postsynaptic cells to presynaptic cells.
Some of it diffuses away and things like that way.
And you have 2 mechanisms to deal with random diffusion. It's going to be taken up by extracellular capillaries in the neighborhood. And also, as we'll be talking about in just a moment, there are glial cells, especially glial cells such as astrocytes, that help to regulate the relative abundance of neurotransmitter near the synapse. Otherwise, you have all this free neurotransmitter just floating about your brain, and it may be good or may be bad, but it's probably not desirable. A lot of drugs do that. A lot of drugs will either block these, increase the rate at which this occurs, or alter this. And so doing, increase the amount of neurotransmitter in your brain. And that can make you happy, it can make you sad, make you other things, make you mean.
Are those two reuptake transporters, are they actively transporting the neurotransmitter, extending energy?
I think if you talk about the mechanism of uptake, that varies. In most cases, these are passive, but they are not well understood. The glial cell definitely has the ability to actively transport. The presynaptic, I think somebody asked about this before, it's a bit more difficult, and you'll see it's almost as if it has the ability to do both, and in some cases in the literature, depending upon the concentration and the signaling. What tends to happen, and we'll talk more about this later. So what is this thing? How would you? Receptor, what is this?
Reuptake.
Well, it's a transport. These are two membrane proteins that have entirely different functions. This cell has the ability, rather like we're sending advisors over to the Middle East right now, this cell has the ability to deploy these on an as-needed basis. And so it seems that the rate of uptake is dependent upon what signals are being sent by the cell. Yes?
What regulates the need for reuptake?
I wish I could give you a good solid answer. I don't think if you were to look at the literature, you would understand. I don't think there's a clear answer to that question. Certainly, the relative abundance of glial cells, the activity of the glial cells, the nature of the brain, or the nature of the CNS, the region of the CNS, the individual circuit. I just think there's so many factors. There's no one answer to that question. And then I think what probably goes wrong, and we're talking about certain types of neuropathology with respect to chemical imbalance and so on. Well, it's probably some derangement or dysregulation that is at least partially responsible. And of course, the drug companies are actively working on ways to modify, modulate neurotransmission. And hopefully by now, from the presynaptic side down through the action potential, through the synapse, you should be able to put yourself in a position of a drug designer and come up with all types of theoretical mechanism by which the neurotransmitter concentration can be increased or decreased, that propagation of the signal along the axon can be facilitated or inhibited. We talked about sodium channel blockers, for example, along the axon. But nature does this too. You can see some examples here. Venom from a black widow tends to increase release of acetylcholine. And again, you have too much acetylcholine in those neuromuscular and autonomic synapses. Botulin does just the opposite. The botulin toxin tends to inhibit release of acetylcholine. Again, think of the consequences of that, the neuromuscular junction and the autonomic nervous system. The bundle toxin prevents the cholinergic receptors from opening. So this is a little bit of so really the receptor channel. So if the receptor channel can't open, the acetylcholine binds to the receptor, but the sodium channel does not open. And then we talked about the curare that tends to affect the motor inflate. It's a paralytic for that reason. Comes from certain South American, it's a South American alkaloid, as I recall. All right. If there are no questions, then we'll go ahead and let me say before we do this, we won't always do this, but I think probably make that the cutoff for the first test.
Perfect.
So that is what we say. Actually, normally, let's assume that everything we cover on Thursday will be covered on the test. And then we're going to have to make an adjustment for the fourth test. That's something we've lost 4 hours on the full test, so we'll have to do something about that. But for this test, just make sure there's no ambiguity. This is the cutoff then for the test return. You may have seen this in the news. This was last week. Did anybody happen to see this in the news? So he's going to do a brain surgery. You might ask yourself, why are they doing this? But does that surprise anyone? That we were going to go and brain surgery and wait there when you play the guitar? If that's surprising, let's take, I have a little bit of a longer video on the same topic. This one's about 5 minutes, but I think we will need a break at this point. So they're discussing the same thing, the same process with a violinist, a famous violinist who went through the same procedure.
To Roger Frisch, the violin is not just an instrument, it's part of him, an extension of his passion for music, beauty, and excellence. Roger's mastery of the violin has put him on the world stage. Few have reached this level of expertise. But not long ago, Roger noticed something wasn't quite right when he pulled the bow across the strings.
When I would draw my bow, I suddenly had a shake in my bow. Now, for most other professions, this would not be a concern. For being a violinist, where your career depends on the stability of your appendages, this was a great concern.
Roger has what's called essential tremor, a nervous system disorder that causes shaking. Concerned, he went to Mayo Clinic where he met neurosurgeon Dr. Kendall Lee.
The amount of tremor he has will be considered pretty mild, but not in the case of a concert violinist.
Dr. Lee and Roger agreed that deep brain stimulation, or DBS, was their best chance at stopping the tremor. It involved placing electrodes into Roger's brain. But there was a challenge to overcome that created a first ever situation in an operating room. You see, because Roger's tremor was so small, it would be difficult for Dr. Lee and his team to tell if the electrodes they were implanting were in the best possible place to stop the tremor.
And so what we needed is we needed a violin in the operating room.
Kevin Bennett heads Mayo Clinic's Division of Engineering. His team worked with Dr. Lee's team to create a violin Roger could play while awake during surgery.
We built and designed a 3-axis accelerometer that we could attach to the bow so that as Mr. Frisch was playing, we could actually see a plot on a computer screen of the movement that really shouldn't be there.
During the procedure, Dr. Lee inserted electrodes into Roger's brain and moved them to the site where the tremor originated. Once there, electrodes fire electrical signals to hopefully stop the tremor. By measuring Roger's movements, the accelerometer told Dr. Lee if the electrodes were in the right spot. There was no guarantee it would work. Thankfully, it did work.
I thought this was a wonderful challenge. This technology could help in this sort of unique situation.
I started playing right away the day I came home from Mayo. I was back playing with my colleagues with Lucid Orchestra 3 weeks later. Without the stimulator on, the tremor is quite pronounced.
You can see the difference. With the click of the controller, Roger can turn the electrodes on and off.
that's as smooth of a bow as I can play right now.
But when it's on, Roger's mastery of the violin is absolutely clear.
I remain very, very thankful every day that I have had the opportunity to have had this surgery. And, I just do not take it for granted.
Back on the world stage to make music, beauty, and achieve excellence.
This is what we're doing here. Let's take a break.